Hello, my name is Alicia Yang, and I am a student researcher in Dr. Albert Kim's lab in the Department of Neurological Surgery at Washington University School of Medicine in St. Louis. Today, I will be presenting my project titled Laser Therapy-Induced Cytokine Changes in a Mouse Glioblastoma Model. First, I will give us some background on glioblastoma and laser therapy. Glioblastoma is an aggressive high-grade malignant brain tumor that is resistant to treatment. In humans, it has an average survival of just 15 months, even with a complex standard of care of surgery, followed by radiation and chemotherapy. Here on the screen, we see a CT scan of a patient who has a glioblastoma tumor and was then treated with surgery by Dr. Kim. In the second row is a CT scan of his recurrent tumor. More than 90% of patients have recurrent tumors even after treatment. And the reason why glioblastoma is so difficult to treat is for the following reasons, and these just list a few of them. And I won't go into detail because the one that I will be focusing on is rampant T-cell dysfunction in the tumor microenvironment. Glioblastoma cells cause the accumulation of tumor-associated macrophages in the microenvironment of the tumor. These macrophages then cause global immunosuppressive reactions in the brain that lead to T-cell dysfunction and T-cell exhaustion. So these are protein receptor markers for cell exhaustion. Common ones that we often see in literature are CTLA4 and PD1. These are often targets for antibodies that have been shown to be very effective in treating other cancers. However, because glioblastoma has low immunogenicity, it is very difficult to treat with immunotherapies. One treatment that has been relatively new in the field of treating glioblastoma is laser interstitial thermal therapy. Laser interstitial thermal therapy, or LITT, is used to selectively ablate tumor cells using MRI and a laser probe. By delivering these pulses of laser, cells are thermally ablated and killed. So I studied this in the mouse model, and on the left we see a diagram of this laser probe, as well as a thermosensor that gives live readings of the temperature in the surrounding area. And that is how I monitor the laser pulses that I'm delivering. On the right is a representative MRI scans from 2 mice that were injected with tumors and then ablated with laser therapy. And we can see that there's a characteristic necrotic core. If we look at this H&E staining of these tumors, we find that in the sham-treated brain in this top row, there are dense tumor cells surrounded by the healthy brain tissue. In the laser brain in the bottom row, we see a necrotic core, and that is showing characteristics of heat and necrosis. So these images are from a publication that I contributed to looking at laser interstitial thermal therapy's effect on increasing blood-brain barrier permeability. We found that this effect extended 30 days past LITT treatment, and so this is one therapeutic benefit of using laser therapy to treat glioblastoma. I wanted to explore the further effects that LITT has on the microenvironment of the tumor, especially given that we can see that there's some inflammation in the brain tissue. And so my question was, what effect does LITT have on the tumor immune microenvironment? And this is a very large question that is very broad. So to narrow it down, I wanted to start somewhere upstream in the process, looking at some of the primary signaling proteins that start the immune response. And these proteins are called cytokines. This is the timeline for my experiment. After 7 days of allowing the tumor to grow within the mice, I performed laser therapy on some of the mice and no treatment on others. Then 2 days following treatment, or 7 days following treatment, I harvested the tumor microenvironment, just a 3 millimeter chunk surrounding the region where I first injected the tumor and then treated the tumors with laser. Then I ran a multiplex immunoassay that was bead-based in order to find the concentrations of the cytokines. This panel included 35 cytokines, 32 of which I was able to find the concentration for across the treatment groups and also the days. Here, I simplistically divided them into pro-inflammatory and anti-inflammatory cytokines. But as you can see, these populations are very diverse. We have colony-stimulating factors, chemokines, interleukins, tumor necrosis factors, and there are relatively more pro-inflammatory cytokines in this panel. But again, these cytokines are very complex. They're often involved in multiple different processes interacting with one another, and depending on their context, they can play multiple roles in mediating inflammation. However, I chose to display this in a more binary way in order to simplify the way that we're thinking about what is happening within the brain. So the first comparison that I wanted to make was just looking at the cytokine profiles of each of the 16 samples. And here that is what we see. These are fractions of the cytokines relative to the total cytokine concentrations of the 16 mice that were harvested, and then also one contralateral brain sample. So here we can see that they are relatively similar across the days and the treatments. However, these 3 mice show quite different cytokine profiles, and this kind of just shows how variable biological samples can be. Generally, across the days, we see similar cytokine profiles between the day 2 post-sham and post-LITT. They're more similar than they are to the day 7 harvested brains. If we look at the total cytokine concentrations, we see that on day 2, the sham-treated brains had slightly higher total concentrations of cytokines versus on day 7, the laser brain had slightly higher total concentrations of cytokines relative to the sham-treated brains collected on day 7. The next comparison that I wanted to make was looking at the individual cytokines and how they related to each other across treatment groups and across the days. So these are just representative graphs of 5 cytokines. I ran ANOVA 2-way tests and also student paired t-tests on each cytokine. Some of these cytokines showed significant differences between the days. For example, tumor necrosis factor alpha showed significant differences between the day 2 and day 7. However, most of these cytokines showed no significant differences with pretty large variabilities within each cohort. The only cytokine that changed concentration significantly across the treatment group on the same day of harvesting the brain was interleukin 18. So here we see a significant downregulation or lower concentration of interleukin 18 in the laser-treated brain as opposed to the sham-treated brain on day 2. So looking at these cytokine concentrations globally, I plotted all of this using a log concentration. So on day 2, these cytokine concentrations are represented by the orange bars, and then the blue bars represented the brains that were harvested on day 7. So from this graph, you can see that there are large variations within each cytokine and even within each cohort. Interleukin 16 sham, we can see a huge error bar. There was no statistical significance between the sham and LITT-treated groups except for interleukin 18. We can also see that pro-inflammatory and anti-inflammatory cytokines are present within the same concentration ranges, meaning that the cytokines above this black dotted line, which are pro-inflammatory, and the ones below are anti-inflammatory, they don't clump in very different ways across this log concentration. So they exist in relatively similar concentrations within the brain. To better compare the cytokine concentrations in laser brain as opposed to sham-treated brains, I normalize all of these concentrations to sham, which is indicated by this red line. And all of the values are normalized to 1.0 so that these lasered brain samples can be represented relative to sham. So the yellow and orange represent the brains that were harvested on day 2. The 2 shades of blue represented those that were harvested on day 7. The darker shades show when the cytokine concentrations were found at higher concentrations in the laser brain as opposed to the sham brain. So from this bar graph, you can see that no cytokines were upregulated consistently across the two time points, meaning that, for example, IL-6 was found in lower concentrations on day 2, but then found in higher concentrations relative to sham on day 7. On day 2, 25 of the 32 cytokines have lower concentrations in the lasered brain. 5 out of the 7 that were found in higher concentrations in the lasered brain were pro-inflammatory. And then on day 7, we see similar numbers, so 26 out of the 32 cytokines that were found at lower concentrations in the lasered brain, and then 5 out of the 6 that were found in higher concentrations were pro-inflammatory. And so generally we see that inflammation is promoted in the lasered brain. The last analysis that I ran to kind of see the population differences between the different treatments and days was to do principal component analysis. The PCA graph on the left shows that the relationships between cytokines is very complex and dynamic. So you see here that the pro-inflammatory cytokines indicated with the green labels tend to clump on the upper side of PC1. However, the anti-inflammatory cytokines tend to be more spread out. And this just underscores the important dynamic relationships between the cytokines and also across the different treatment groups. In the left graph here, we see that time point seems to provide a stronger association between the cytokine profiles than the treatment groups. We can see that because the sham and laser-treated brains don't tend to clump together. Rather, it's more the populations tend to clump together based on the day that they were harvested after treatment. From these results, we're able to show some trends in cytokine changes after laser therapy. The 3 main conclusions are shown here. First, that cytokines increase with time after surgery with a slightly larger change in total cytokine concentration in the laser-treated mice as opposed to the sham-treated mice. This points to the fact that perhaps laser has an effect on increasing the number of pro-inflammatory cytokines that are being released in the tumor microenvironment. Secondly, that the fraction of cytokines in the tumor microenvironment changes over time, and so time plays a significant role in determining what cytokines are present. Lastly, although we did not see significant changes in cytokine concentrations, LITT did play a moderate role in increasing pro-inflammatory and anti-inflammatory cytokine concentrations in the brain, either 2 days or 7 days after therapy. The challenge in drawing strong conclusions from this data is that our cohorts were really small, and so I wasn't able to run multilinear regressions. So in the future, I will be increasing cohort sizes to clarify trends and track changes in tumor microenvironment over time and also across the treatment groups. I will be corroborating these findings with immunohistochemistry staining of T-cell localization in the lasered brain and also running flow cytometry to measure other immune cell compositions. I also started creating a slower growing tumor mouse model with a lower cell injection number over the summer in order to allow the mice to mount a natural immune response before treating them with laser therapy. Lastly, and this might happen further into the future, but the idea is to be able to test laser therapy in combination with other immunotherapies in order to increase the survival of mice with glioblastoma. I want to end by thanking all the people who have helped me conduct my independent research this summer. First is Dr. Albert Kim for giving me the opportunity and the freedom to explore and grow over the past 2.5 years. He is the perfect model of a compassionate physician, but also a hardcore, dedicated researcher. And I want to emulate these values, and I'm so grateful that he gave me the opportunity to really improve my skills as a researcher. I want to thank all the prior and current members of the Kim Lab and Yano Lab who have helped me develop the skills that I needed to conduct this research. I want to thank Dr. Diane Bender for helping me run the immunoassay and BioServe for funding my research and allowing me to share it. Thank you for listening, and if you have any questions or comments, please email me. Thank you.